So now we'll have a lecture about cell type prediction, and we invited Ahmed here from the Netherlands because we know he's a good teacher and that his group has done an excellent paper on comparison of different cell type prediction methods for single cell data that I can recommend you to look at. So go ahead, Ahmed.
Yes, thanks a lot, Asa, and thank you all for being here. Indeed, I work in Leiden in the Netherlands, at the University Medical Center, and I'm also part of the bioinformatics group in Delft. And I will tell you something about cell type prediction. I want this to be as interactive as possible, so if you really have any questions, just jump in and stop me. Okay, why are we interested in this? So my lab is really interested in understanding a bit what are the effects of genetic variants on diseases and specifically on brain disorders. We have this cartoon here from the consortium where we, say that over the past, 20 years or so, GWAS studies have been pouring these genetic variants on biologists. But then studying the effects of these variants is really difficult because each variant has a very small effect. So you really cannot take one variant and design a mouse model easily to study the phenotype. So one way to go about this is to basically study the function of these variants before you pass them to the biologist. So you say, okay, we think this variant is mostly affecting this specific cell type. So in Alzheimer's, most people now focus on microglia because we know that this is probably the cell type where the variants for AD are mostly exerting their effects to generate the phenotype that we observe. So this is a bit the approach that we take. So we take these genetic variants, we rely on transcriptomic data. Earlier, we relied a lot on these atlases that map transcriptomic data across different brain areas or development stages. But now, of course, we rely a lot on single cell data as well. And of course, with single cell data, there are a lot of technical challenges. So today we're talking about one, but throughout the week, you have also seen why this is an interesting problem for computational biologists in general. This is just an example to show why studying cell identity is actually important. So this is a preprint from the Allen Brain Institute where they study cell types in the motor cortex across different species. And what you see here is that you have different cell types in human, in macaque, and in mouse. And what they show here is that these are conserved marker genes that you can find in the same cell type across the different species. And here you see these are more human-specific markers. Sorry, this is a marmoset, not a macaque. And these are marmoset-specific markers, and these are the mouse-specific markers. To do this analysis, you really want to make sure that what you call a somatostatin neuron, for instance, in human is exactly the same cell type in the other species as well. And there comes the problem, because you might rely only on the markers like you do here, but then markers might not be very well conserved. So what is really the same cell type to start with comparing that? There are a lot of ideas, and there are a lot of philosophical ideas on what do you actually call a cell type. Does it have to have the same marker? Does it have to have the same developmental origin? And does it have to have the same location in the tissue? We rely only on transcriptomics in this talk. So we only rely on the observed gene expression pattern. So we're not considering location or transcription or epigenetic status of the cell or lineage or whatever. So we're only talking about transcriptomics. Okay, so how do we identify cell populations? I think you did this already in the course so far. So you take a count matrix where we have observed the counts for specific genes across a different types of cells. And at the end, you want to end up with annotated cell types, right? The way to go about this is that you do clustering. So you group your cells into groups of cells that have homogeneous or the same expression pattern, more or less. You start to overlay markers. So these can either come from your prior knowledge about the tissue. So if you're looking at an immune system, then you would look at the canonical markers that would identify for you the T cells or the B cells. Or you can do differential expression where you say, okay, I don't know what this cell type is. So what are the markers that are differentially expressed between this group and the rest of my cells. And then if you get markers that can give you an indication what these cells are. And this has been working really nicely. So here a bit of a shameless plugin. We also have a tool called Cytosplore Transcriptomics that we just released that allows you to do this very interactively. And there are many other tools, by the way, and I'm not claiming this is the best, it's just a way to do it. Just so you can create your TSNE maps, you can overlay markers, you can, encircle a group of cells, find differentially expressed genes, and so on. And this works really well for most people, but there are problems. So if you have ever done this, or if you will do it also in the future, this is a very time-consuming process, especially if you want to annotate your tissue at a deeper level, that you want to define really small or specific cell populations. It's highly non-reproducible, because if you do it with two people in the same lab, even, you will definitely get different annotations of the same cells. Maybe you would agree at a certain level, but at deeper levels, people tend to disagree. There are a lot of subjective arguments or decisions that you have to make, whether this marker is indeed expressed or not, and whether it's unique for this population or not, and so on.